Okay, good afternoon. The next lecture that we're going to talk about is metabolism. And this is a topic that should not be new to you. This is something that should have been covered in either biology or anatomy. Specifically when we talk about the pathways like cellular respiration, dealing with glycolysis, TCA cycle, electron transport chain. These are all topics that, are sort of topics that should be covered in prerequisite courses for this course. We will get into this again, and if you've looked at the review sheets that I've posted, you can see that I sort of have given you a few different views to sort of look at this material and hopefully study this material. You do need to know these pathways for micro. You will be tested on these pathways. So please make sure that you do spend some time with this. This is not an easy topic. And that's partly the reason why you'll notice that, we finished this module is pretty much just viruses, as well as metabolism. Okay, and you'll see that there's not a lot with this particular excuse me, with this particular chapter. For the recorded lecture, I am probably going to break this up into 2 or 3 different recordings, just because there's actually 3 different areas that are sort of covered in this chapter. The first recording, which is the one we're currently in, will sort of be a background on metabolism and looking at enzymes and things like that. The second will deal with cellular respiration, and the third recording will be fermentative pathways or anaerobic respiration. So looking at the different types of chemical reactions that can occur in a cell, you should be familiar with these 2 terms, catabolism and anabolism. Catabolism is the breakdown of complex molecules, and when you break down molecules like that, you are breaking bonds. And when you break bonds, you typically release energy. Anabolism is the opposite. This is when you take smaller molecules and you form bonds between those 2 molecules. And this usually requires the input of energy. And here we can look, we take a glucose molecule, which is usually the example that's used for cellular respiration. And when we break it down, we end up releasing CO2. But as we break the bonds holding the carbons together, we end up releasing energy, which ultimately ends up on an ATP molecule, which the cell can then later use for other things. Anabolism, on the other hand, would be taking smaller molecules such as amino acids and forming peptide bonds between those amino acids and making proteins. And that requires the input of energy. Now, metabolic pathways, they are sequences of enzymatically catalyzed chemical reactions. What this means is that you're going to have a series of reactions that occur in a metabolic pathway. It could be, 1 or 2 reactions, or it could be as many as 10, 12, 20 reactions. Depends on the pathway. These reactions are going to be catalyzed by an enzyme. Okay, and each reaction of the pathway most likely will be catalyzed by a different enzyme. Now, metabolic pathways are determined by their enzymes. What this statement is saying is that in order for you to, say, do glycolysis is a pathway that actually has 11 different enzymatic steps, which means there are 11 different enzymes to complete glycolysis. And in order to complete glycolysis, you actually have to have those 11 enzymes present in the cell. If you're missing even just one of those enzymes, you might not be able to actually complete glycolysis. What determines what enzymes a cell will have ultimately comes down to its genes, for the simple reason that enzymes are proteins, and because enzymes are proteins, in a cell, a protein gets made from a genetic transcript, which is a gene. Okay, so what a particular organism is able to metabolize as far as metabolic pathways are concerned is all dependent on its, genetics. So one of the questions that you'll see, and I guarantee you'll see this question on a test, is what is an enzyme? How does it work? And a lot of people will come back and say it speeds up chemical reactions. That's the outcome of an enzyme, is that yes, it does speed up chemical reactions, but the question is, how does it work? And the answer to that is what you see here, activation energy. What an enzyme does is it lowers the activation energy for a enzyme, okay? I'm sorry, the enzyme lowers the activation energy for a reaction. Now, what does that mean? It essentially means that you require less effort for the reaction to actually occur. And there's what we call the collision theory. And what I would like you guys to imagine is picture you have two molecules sort of bouncing off the walls in a room. And the chances of those two molecules coming together and actually forming a bond with each other is very slim to none. Not impossible, but slim to none. What an enzyme does, now picture that you have an enzyme, and the enzyme is sitting in the middle of that room. The enzyme, because of its unique structure, due to the different types of amino acids that it's composed of, will actually sequester. Sequester means to attract the substrates, which would be those two molecules floating around the room. And now those two substrates are going to be attracted to that enzyme, and they're going to bind to the enzyme. So that what you then have is you now have the two smaller molecules that are on the enzyme. Now the likelihood that those two enzymes will form a bond is very likely. So what are enzymes? Enzymes are catalysts. Catalyst is a chemistry term. It's something that's typically used to speed up chemical reactions. Now, enzymes are taken into a whole nother definition because they are what we call biological catalysts. And they are biological catalysts that are made up of primarily proteins. Enzymes act on a specific substrate and they lower the activation energy for the reaction to occur. Because they are made up of proteins, they are subject to denaturation. Denaturation means that the protein can unfold, and when the protein unfolds, what happens is it loses its function, which could affect metabolism within a cell. That's why temperature is very important in that regard, okay? Now, when we look at an enzyme, and I'm going to go to the picture here. Here we have an enzyme. Here we have our substrate. If you note that this is a pretty ornate fit here. This is like a puzzle piece fitting in. And that was intentionally done. What that's meant to do is it's meant to signify specificity, that a substrate binding to an enzyme is a very specific event, and that specificity is very, very unique. And if there's any alterations in that specificity, that could affect the ultimate binding of the substrate. The other thing that I want to point out here is, when the reaction occurs, and let's say we're splitting this molecule into two, so this would be a catabolic reaction, and we end up releasing the products. The products will never contain anything but what entered in as a substrate. And everything that goes in has to come out. The one thing that I want to also point out here is if you look at the products, you see that there's no green in the products, meaning that the enzyme never, ever, ever becomes part of the products. It's just there to facilitate the reaction. Okay, this is some more background on some enzymes. Naming of enzymes. When you look at the names of enzymes, actually look at the names of the enzymes. Maybe look them up. Look at what that name actually means, because it's probably going to tell you what it's doing. We're going to see some enzymes that get produced and secreted by bacteria. For example, collagenase. Collagenase is an enzyme that is secreted by some bacteria. And what collagenase does is it breaks down collagen. Now, where might you find a lot of collagen? Skin. You might have that enzyme along with keratinase, and those two enzymes are being secreted by bacteria, and that could actually cause, breakdown of that surface of the skin, making it more invasive for that bacteria. More in lines with metabolism, you might have an enzyme that's called a decarboxylase. Decarboxylase means to remove carboxyl carbon. So a decarboxylase enzyme is going to end up removing carbon. And you'll actually see, for example, pyruvate decarboxylase, which is part of the one of the pathways respiration, which is involved in removal of carbon. So again, look at the names. Look up some of these names that you come across when you look at these pathways, and make a little note of them, because the names will actually tell you a lot of information. We'll have cofactors and coenzymes. Cofactors are non-protein components. A coenzyme is a cofactor, but it's an organic cofactor. Okay, it's most likely a vitamin or something. The coenzyme or cofactor is just as important to the enzyme as the amino acids that make up that enzyme. And a lot of times, substrates might not be able to bind if it does not have the coenzyme or cofactor present. Some very common enzyme components that assist enzymes are what we call electron carriers. And you're going to see these guys as well. And I'm sure you guys have seen these guys before, especially, for example, NAD and FAD. They are electron carriers. Their job is to pick up electrons when they get released from a reaction that is breaking a bond, and transport those electrons to, say, the electron transport chain, where they will then be further processed. But we'll come back and talk more about those a little bit later on. Some factors that can influence enzymatic activity. We have temperature, pH, substrate concentration, and various inhibitors. So with temperature and pH, one of the things that can actually happen is you can end up with denaturation of the protein. If the temperature gets too high, essentially what happens is the bonds holding the protein together melt away, and the protein unfolds. An unfolded protein is a non-functional protein. With pH, any extremes in pH, whether you go too acidic or too basic, so this is going on either end of the spectrum here, you will break those bonds, the protein will unfold, and again, an unfolded protein is a non-functional protein. Here we can see activity, enzymatic activity with temperature. As we increase the temperature, we start to see an increase in activity. But as we get to this 42-degree mark here, what happens is you see an immediate drop-off of the activity of the enzyme. And this is because what happened at 42 degrees is the enzymes denatured and we lost activity of the enzyme. Same thing with pH, too acidic, too basic, we lose activity. Okay, they just can't deal with that. Enzymes can also be saturated out. What this means is that if you have two enzymes in a cell that are able to start the process of glycolysis, what that means is you can only process 2 glucose molecules at a time. Which means if you have, say, 100 glucose molecules, you're not going to move any faster. You're only going to process 2 glucose molecules at a time. Now, what can happen is cells do respond to their environments. So in order to process more glucose, because there is more glucose, what you can do is the cell can make more enzyme. And if the cell makes more enzyme, you can actually increase the amount that you're able to process at any given time. Inhibitors. Inhibitors will inhibit enzymes. We have 2 types of inhibitors. We have competitive inhibitors, and you can see that guy in yellow right here. So normally we would have our substrate binding to the active site. And here, because the inhibitor bounds, the substrate's not able to bind. Now, why is this important in micro? Well, most antibiotics actually target things like enzymes, especially enzymes that are responsible for synthesis of the cell wall. I know I mentioned a little bit early on that antibiotics target cell walls. And the way they do that is either by this competitive inhibition, or you'll see in a minute, non-competitive inhibition. So if this was an antibiotic that is binding to this active site, this enzyme can't function because the substrate can't bind. Now, if this is an essential reaction that has to occur for the cell to stay alive, that's a problem for the cell, okay? And this could lead to death of the cell. If you do have a competitive inhibitor, you want it to bind better and faster than the substrate itself. We also have what are called non-competitive inhibitors or allosteric inhibitors. And what happens here is, if you look, we now have the non-competitive inhibitor, right, binding. But what that did is, when the non-competitive inhibitor, so it's binding someplace else besides the active site. But look what happened, what it did to the active site. When this ended up binding, it changed the shape of the active site. By changing the shape of the active site, you still get the same result in the fact that the substrate no longer fits and cannot bind. If the substrate cannot bind, it cannot continue. Okay, the reaction cannot continue. Now, most antibiotics are actually non-competitive inhibitors. Okay, they are not competitive inhibitors. And a problem is that because most antibiotics are non-competitive inhibitors, we have a little bit of a problem. And that is the ability to develop drug resistance. And the way that that happens is drug resistance is a result of a DNA mutation. And in this case, the mutation might occur in the gene that makes this enzyme. And let's say that that mutation leads to an amino acid change. And now the amino acid is changed, and the new amino acid, instead of forming this binding site that we see here, if you look over here on the left, you see I'm drawing what the new amino acid might look like. Okay, and now this new amino acid sticks out like that. Well, what's going to happen to our inhibitor? The inhibitor is not going to be able to bind now because it's not going to fit in there. And if the inhibitor can't bind and fit in there, that means that you're not going to affect, this shape right here of the active site, which means that the substrate can continue to bind. So what you've essentially done is, by doing that mutation, you have blocked the binding of the drug because it doesn't fit anymore. And now the original substrate is still able to bind and the reaction occurs. Good for the bacteria, bad for you, okay? Now, and I said most antibiotics are going to be that non-competitive inhibitor. Now, if you had a non-competitive inhibitor and you did the same thing there, the problem is that if you have a mutation that's now sticking out like this, not only would the antibiotic not bind, but the substrate won't bind. So you still get the same effect. So mutations don't work very well with competitive inhibitors. But the problem is that most antibiotics are non-competitive inhibitors where mutations work very well. And it's just easier to find a drug that's going to bind somewhere else on this enzyme versus this very specific area of the active site. That's why most antibiotics are actually non-competitive inhibitors. So again, I hope this gives you a little bit more of an appreciation on why studying these things is a little bit important for micro, because it really gets down to the mechanisms of actions of, say, how drug resistance is able to form. Okay. And just bear with me for 1 second. Let me go back to my little arrow here. Oops, sorry. Okay, so now that we've finished talking about enzymes, we're now going to get into some types of reactions. And some background of one of the major types of reactions that you'll see with these metabolic pathways is what we call oxidation reduction reactions. Oxidation is the removal of electrons, meaning that you have broken some type of bond and you have released electrons. Reduction is the gain of electrons. So you're forming some type of bond or something is picking up those electrons. Unfortunately, or fortunately, however you want to look at this, the way that our universe is set up is that energy is neither created nor destroyed. It is just transferred from one form to the next. The way that that applies to these for this is that whenever you release electrons, something has to pick them up. You just can't have free electrons out there. So in an oxidation reaction, when you complete oxidation and you end up releasing the electrons, something else has to come in and pick up those electrons. And when that's something that comes in to pick up those electrons, what that actually is the reduction reaction. Because these two reactions are linked together, and they are, permanently linked together, which means when you do one, you have to do the other. We call these redox reactions. And, if we sort of look here, if this was an organic molecule such as glucose and we split glucose into half, we end up releasing electrons. And what you're going to find on the pathways that you're specifically going to study with this is that when you release those electrons on a pathway, they're most likely going to get picked up by NAD. Okay. And what we then have is our oxidized molecule, our split molecule, now with a reduced NAD, which is NADH. And what will happen is now this NADH will then travel to the electron transport chain, where it then will get oxidized and release the electrons into the electron transport chain. Hence this being just a carrier. It's picking up the electrons on the pathway and then transferring them to the electron transport chain. Okay, so initially it got reduced because it gained the electrons. And then when it gets to the electron transport chain, it will get oxidized where it will release those electrons. Okay, so the last topic that I want to cover for this first recording is the generation of ATP. Now, ATP is generated by the phosphorylation of essentially adenosine diphosphate to make ATP with the input of energy. And there's going to be three ways to make ATP. But before we get into the three ways to make ATP, I want to first talk about ATP. You should know the structure of ATP. That is something that was covered in Chapter 2. Make sure you know what ATP is composed of. I'm guaranteeing you're going to see a question on this. Okay. So it's an adenosine molecule, which is a nucleotide, plus 3 phosphate molecules. Okay. So ATP can be generated 3 ways. And this goes for any living cell. There's only 3 ways that you can make ATP. Most cells will only do 1 or 2 of these. They can't do all 3. And you'll see why in a second. The first way to make ATP is what we call substrate level phosphorylation. Substrate level phosphorylation is when you take a molecule such as ADP and you take another molecule that has a phosphate that it's able to donate, and an enzyme takes that phosphate off the one molecule and puts it onto ADP to make ATP. This is essentially a transfer reaction. Despite its name of sounding very complicated, it's not. It is a transfer reaction. That's it. What happens is, let's take this molecule, for example, here. We have a 3-carbon molecule, right? And this 3-carbon molecule has a phosphate. Here's that phosphate right there, okay? Then we have ADP right here. The arrow is going to designate our enzyme. I'll just put a little E here, okay? Bear with me, it's very difficult to write with a cursor. And what's going to happen is we're going to chop this bond here, break this bond, and we're going to transfer this phosphate to the ADP molecule, okay, making a 3-carbon molecule that's now absent of the phosphate plus ATP. That's it. It's a transfer reaction. This does not require oxygen. We don't see oxygen anywhere in the equation here. So theoretically, this form of ATP production can occur both aerobically and anaerobically. Oxygen is not a factor, so it can occur in either case. Because oxygen is not a factor, this is something that we will see in fermentative pathways. The second way of making ATP is called oxidative phosphorylation. Oxidative phosphorylation is when electrons are transferred from one electron molecule carrier to another along the electron transport chain on a membrane, ultimately releasing energy to generate ATP. Essentially, the electron transport chain is where the process of oxidative phosphorylation occurs. If you look at the name oxidative phosphorylation, one of the things that you should be able to automatically infer is that oxygen is required. It's kind of part of the name. But essentially what happens is the process of ATP generation on the electron transport chain by taking the electrons and making ATP is oxidative phosphorylation. So when you see the electron transport chain, it is oxidative phosphorylation. And this is really what defines cellular respiration, okay? The third way to make ATP is what we call photophosphorylation, and it actually has another name, and the other name is called photosynthesis, something that you probably talked about in your early biology days, however long ago that was. Photosynthesis is a little bit different than cellular respiration. Essentially what photosynthesis is you're taking light energy, converting it to chemical energy. That chemical energy could be in the form of sugars, or it could be in the form of ATP, okay? So photophosphorylation is taking light energy and converting it into chemical energy such as ATP. And that's why I said, most cells will either do one or two of these pathways. So for example, if you look at human cells are only able to do substrate level phosphorylation and oxidative phosphorylation. But if you look at a plant cells can undergo both substrate level phosphorylation and photophosphorylation. So it's kind of one or two pathways. It's very rare that you'll have a cell that can do both cellular respiration and photosynthesis. So as we dive into these pathways, we're going to see substrate level phosphorylation. You might see on some of the review sheets that I've gone through the pathways that you'll need to know. I abbreviate substrate level phosphorylation as SLP, and that's how those ATPs were made. You'll see oxidative phosphorylation. That's really the bulk of where you get most of your ATPs through cellular respiration. Oxidative phosphorylation on the review sheets is abbreviated by OP. We're not going to get into photophosphorylation. Most microbes are either just going to do those substrate level or oxidative. But I just wanted you should be aware of the actual three ways to make ATP. And I would not be surprised, I know it's one of your open-ended questions on your lecture assignment is what are the three ways to make ATP? You want to list those three ways, but you also want to describe what's happening with those three ways, okay? Don't just list them, put a little description next to them, because I would not be surprised if you saw an open-ended question on your second exam that says list and describe the three ways to make ATP. Hint, hint. Okay, so that brings us to our metabolic pathways, and that's where I'm going to end recording 1. And I will pick up on carbohydrate catabolism with recording number 2. Okay, probably tomorrow. Enjoy the rest of your day, guys.